speed with which the vaccine is actually developed in the laboratory with all the new techniques, it still has to undergo some rigorous clinical trials. And there is that very essential requirement, which may be painful in terms of time delays, but absolutely important in terms of making sure that the vaccine is what we want in terms of efficacy, safety, and if possible, long duration. So we have trials in humans, essentially 3 phases, but there's also a phase 4, which we can discuss later. The first is the phase 1, which in a small number of volunteers, you inject the vaccine and then find out whether it is safe or not. The immediate thing is there safety or not? Then in the phase 2, you increase the number of volunteers and then administer the vaccine again. In this case, you're looking for not only safety, but you're also looking for the right kind of dosage. What are the kind of antibodies that are being produced? Are they being produced in adequate quantities? Are they neutralizing antibodies? And is the dose something that has to be increased if it is insufficient? So essentially, you're partly looking at initial promise of safety, but also looking at the response in terms of efficacy for dose adjustment. But still, the numbers are not large enough and the process is not objective enough for us to judge how effective the vaccine is. So that is where we get into phase 3 trials, which is a proper randomized clinical trial, which usually involves thousands of people, because these are healthy people who are at the risk of exposure to the virus, and we want to prevent the virus from attacking them and causing an infection, even if they are exposed to the virus. And therefore, we require a large number of people to prove both efficacy as well as safety. And this is absolutely critical. And now, this is because, and then secondly, we also require, if possible, some idea of the duration of protection. Though a phase 3 trial may not be able to give you a full evidence of the long duration, at least apart from antibody production, if you can also demonstrate that there is a good T cell activation, then you're reasonably assured that the duration may be a bit long and not going to disappear in a few weeks or a couple of months. So those are important. But then there is also a phase 4 study. In a phase 4 study is what we call post-marketing surveillance. That means after phase 3, if you have passed the safety and efficacy tests, then the regulators usually allow it for use. They license it for use. And that, after getting that regulatory approval, when the virus is now being used much more widely, still you have to actually record the experience as much as possible across the population, because the numbers are now increasing. Some side effects you may not have noticed in the trial, where the sample size may not have been adequate, may come forth, and then you may find out it's not as safe as it was believed to be. And secondly, you'd also get a better idea of the efficacy, because now you'll be able to record much larger number of infections in the population, and then find out what was the level of protection in the vaccinated people versus the unvaccinated people. So that is the real world experience that we gather in the phase 4 trial. But as I said, it is phase 3, which is the real test for regulatory approval.